Hello, everyone. So, welcome for today's webinar on the sixth webinar series of the Asian Solanaceous and Cucurbits Roundtable. As you all know, today's theme is genome editing and Asian regulatory landscape. So, before we continue with the webinar, I would just like to go through a few housekeeping rules. As you know, all the attendees are not allowed to open their microphone or the camera. So at the bottom of your screen, you can see the chat and the Q&A option. So you can use the chat box for technical issues, comments, or remarks, and you can use the Q&A box for asking your questions. So in the Q&A, as you can see in this example, you will see the like button. So if you see a question which is also interesting for you, you can put your like on that option, and we will help to answer the most liked questions after each session, and then the remaining questions will be answered later via the speakers. So with that, I would also like to thank our today's sponsor, which is Foundation of Agri-Tech Commercialization and Transfer. You will just hear a little speech about the sponsor, and then we'll start with the webinar.
The Korea Seed Expo 2021 will be held online from October 6th to November 2nd. We hope many people will show a lot of interest in the Korea Seed Expo 2021, which will bring together Korean seed, agricultural machinery, and other agricultural material companies. Come join us to find out about the latest varieties, genetic resources, and field crops from these seed companies, which will be featured in demonstration field of the K-Seed Valley. Visit koreaseedexpo.com.
All right. With that being done, I would like to thank everyone, all the attendees today for joining this webinar today. I would also like to thank the R&D Advisory Group for arranging this webinar series, and our respective speakers for their speech today, and the Secretariat as well. Without taking more time, I would like to invite Dr. Divya from R&D Advisory Group to introduce our first speaker and start the webinar. Dr. Divya.
Thank you, Muazzim. A hearty welcome to all the participants for the 5th APSA ASRT Expert Crop Improvement webinar. Today's theme is Crop Improvement through Genome Editing and the Asian Regulatory Landscape. Genome editing is revolutionizing plant science and its application in agriculture. In its simplest form, it can generate specific genetic variants that are indistinguishable from naturally evolved variants when compared to transgenic technology, which can only introduce biologically non-existing foreign genes to the original organisms in order to tailor the species with new traits. Therefore, the use of gene editing technology can be fast, accurate, and without the introduction of exogenous DNA fragments. The legislation and regulation of genome-edited plants in many countries is similarly evolving rapidly to adapt to new technologies. So in today's webinar, we summarize and provide an assessment of the current status of this rapidly evolving regulatory landscape with a focus on recent policy developments in Asia Pacific. For today's session on gene-edited tomato for human health, it's our great pleasure to invite Dr. Hiroshi Ezura from University of Tsukuba, Japan, who has engineered the recently developed high GABA tomato utilizing CRISPR-Cas gene editing method. Dr. Hiroshi Ezura is Director of Plant Innovation Research at University of Tsukuba, an experienced plant molecular researcher with extensive experience of gene editing in Solanaceous and Cucurbitaceous crop. Over to you, Dr. Hiroshi.
Thank you very much for the introduction. And I'm going to share my slide. Okay, can you see my slide?
Perfect, sir.
Yes.
Okay. Thank you very much for the introduction. My name is Hiroshi Ezura, and from the University of Tsukuba. And I'm also the CTO of the Sanatec Seed Company, which is a venture company from our university, which is a venture company for commercialization of gene-edited crops, not only in Japan, all over the world. Okay, so today I'm going to introduce the gene-edited tomato for human health. And mainly the high GABA tomato as a case study of the development of application of gene editing for crop improvement and also the commercialization. And today's my talk contains 4 topics, and one is a background of development, and the second of the proof of a concept research on the development of high GABA tomato by gene editing technology. And the third, the development of a commercial high GABA tomato and its commercialization in Japan so far, and then the take-home messages. Okay, so as a background of my presentation, I would like to introduce our Japanese national project, which is name is Cross-Ministerial Strategic Innovation Promotion Program, just abbreviate it SIP project, which is supported by Cabinet Office of Japan, and which is funded for 2014 to 2018 for 5 years. And this means that our Japanese government strongly pushed this application, application of this new technology for the improvement of agriculture products. And then in this the project, I lead the establishment of new breeding techniques, and which include the almost more than 50 Japanese institutions for the application of these gene editing technologies. The consortium is I established 4 consortium, Group 1 and Group 1, which is developing a new generation breeding technology. That means that new gene editing technologies. And for the Group 2, which is establish a platform to find a target gene on gene editing. And the Group 3, which is a group to demonstrate the usefulness of this gene editing technology for the improvement of agricultural products, and which include like a vegetable, like tomato, and also rice and potato, and tuna fish. And Group 4, which is a quite unique consortium, which is a social implementation consortium. In this project, we simultaneously started the social implementation, or we decided to provide the information at the early stage of development to the consumers. So that is very help to accelerate the commercialization of these products from this project. And then we spent 5 years, and then now we have several potential gene edited crops and fishes developed in this project, such as super high yield rice. By gene editing, engineering one genes to introduce the mutation in one genes, we could increase the yield, 20% of yield compared to the original cultivars. And also the potato with a low natural toxin, and that will be going to be commercialized in a second crops, maybe in Japan. We in this project, we knock out the natural toxin accumulation in potato, like solanine and chaconine, by using a gene editing technology. And also we try to apply this technique to the fish improvement, and tuna fish is very famous for Japanese diet, and then we try to improve the tuna fish for easy aqua cultivation. And also my group is focusing on the application of gene editing technology for the mainly a vegetable crop, which include tomato, as you can see here. And then from the next slide, I would like to introduce what we did in tomato in this project. Okay, before introduction, what we did in the tomato, and I need to maybe introduce the current situation of in Japan, and what is the problem, and which have to be solved. And you can see that, and many people say who are related to the food security, and now the world population is going up to almost 10 billion in the 2050. So that's why we have to increase the food production and also develop a technology to sustainably produce a food. So this is the world situation. But in Japan, as you can see here, our population in Japan was a peak at 2010, almost 10 years ago. And then after that, our Japanese population is getting reduced, as you can see here. And then according to this calculation, we are going to lose 30% of Japanese population in the next 30 years. So this is a very, very big problem. And this makes many issues we have to be overcome in the next 30 years. Okay. So we saw that this new gene editing technology can be give us some solution to improve this, overcome this situation. And in this slide, you can see the structure changes in Japan. And in 1975, we have a lot of young generation, as you can see here, like a pyramid shapes. In the 2000, we have a lot of middle ages population. And now we are 2021, and then we have a lot of aged population. And the 2050, we have a lot of aged population. We're expecting more than 40% of Japanese population will be 65. So in Japan, we are retiring at 55. But anyway, so depending on these population changes, we have a lot of lifestyle-related diseases in Japan, like high blood pressure and obesity and diabetics and so on. And so, and of course, we are taking care of those lifestyle-related diseases by the medical care, but it will not be enough to fully overcome our current or future situation for this in lifestyle-related diseases. Then maybe alternative solution is a health promotion via daily food. So that's why we are focused on the improve the quality or nutrition of the agriculture products using a different kind of technologies. And gene editing technology is one of those technology. And in my group, we are studying the fruits crop, including like a Solanaceous crop, like a tomato, and also the Cucurbit crop, like a melon, and some other, like a cucumbers. And for those crop, we are trying to understand, we are spending a lot of our time for basic research to understanding the molecular regulation of important breeding trait. And then we are trying to, we already identify several key genes which can regulate the shelf life and the fruit setting and like a personal caloric, and also sugar accumulation and nutrition like GABA and the lycopene and the beta carotene in the tomato roots. And then we know that we have information of what kind of mutation can in genes can improve those important breeding traits. So then I spend a lot of time, my laboratory spends a lot of time to this kind of basic research to understanding the genetic regulation of the important breeding trait. So then new techniques is appeared, gene editing technology appeared after 2000. Earliest one is the early 2000, and then more convenient one, which CRISPR-Cas9 is released in 2012. So then we decided to apply this gene editing technology for the improvement of such an important breeding trait. And from here, I would like to introduce a proof of concept research on the development of high GABA tomato by gene editing technologies. Okay, in this slide, I would like to what is a GABA. GABA is a very famous in Japan now, but maybe not familiar in other regions of the world. So GABA is a gamma aminobutyric acid, which we abbreviate like a GABA, and which is a non-proteogenic amino acid and found in all animals, plants, and bacteria. and those GABA is involved in biotic stress responses in those organisms. But for human, GABA has a function as a neurotransmitter. So okay, so then that's why it can reduce the blood pressures and also induce relaxation, as you can see here. Because of this reason, then many food companies in Japan, they use GABA as a food additive and then they produce different kind of health promoting functional food. Here you can see some example, and some companies which mix it with chocolate, and also then some company prepare some beverage with a GABA to improve sleep qualities. So that's why the GABA is a very, very famous functional ingredient in Japan so far, which is like a vitamin C in Japan. So from this reason, from this reason, my group is studied the GABA metabolism in higher plants, especially in tomato. And here you can see our conclusion, how we got the information how GABA synthesized in the tomato fruits and also degraded in this. And also we identified the enzymes related to GABA metabolism and also genes coding for these enzymes. And in conclusion, we know that this GABA enzyme is a key enzyme to regulate the GABA content in the tomato fruits. And here you can see in the basic research almost more than 10 years ago, and we got the information that identify the genes coding GAD enzyme, where at the time we identified 3 GAD genes, and also we identified 3 GABA-T gene, which is enzyme degraded GABA, and also another degradation enzyme SSADH1. And then through the basic research, we identified that this GAD3 is key genes regulating the GABA accumulation in tomato fruits. So that's why we targeted to engineer and understanding to GAD3 gene more deeply. Okay. And from our study, biochemical study, and we know that there is some autoinhibitory domain at the C-terminal end of this enzyme. Then if we delete this C-terminal end, which called autoinhibitory domain, AID, so then this enzyme show a more higher enzyme activities. So from this result, we thought that if we can delete this C-terminal end from the GAD enzyme, which can show a higher GAD enzyme without this C-terminal end show higher activity in the cells, and that's resulting in the higher GABA accumulation. That is our speculation. Then we used the CRISPR-Cas9 system to delete the C-terminal end of this enzyme genes. Then we use a CRISPR-Cas9 system, which is developed by reported on the 2012 from American group. And then by using this CRISPR-Cas9.
Dr. Hiroshi, sorry to interrupt. Your slides are not moving actually.
Really?
Yes. We are still on the GABA slide. What is GABA? We are still on that slide.
Oh, really? Okay. So
yes,
how should I? Can I retry the sharing slide? Okay.
Yeah, now we see the regulation of GABA content slide. Could you please try to go maybe back or forward for one slide, just to make sure?
Okay. How
Yeah,
about,
okay. Yeah, now it's working. Thank you.
So can I start from this slide?
Yeah, okay, sir.
Okay, so regulation of GABA content in tomato fruits by CRISPR-Cas9. We use a stop codon mutation at the end of this, just in front of AID domain, and then that's by a CRISPR-Cas9 techniques, and then that induce a stop codon in the genes, and then that's resulting in the deletion of this AID domain. As you can see here, this is a line which we created by this gene editing techniques, D53 and the I1 and the D60 I and 1, and then you can see the GABA accumulation in the fruits. Wild type is the original cultivar, and then this other bars show the GABA content in the CRISPR mutants. And as you can see here, the GABA accumulation is completely increased compared to the wild type, which increased to 10 times to 15 times maximum. 15 times maximum, and so. This slide demonstrates that our strategy to effective to increase the GABA content in tomato fruits. And the next experiment, as you know, the tomato has been used as a F1 hybrid. So then we created a F1 hybrid with a high GABA tomato created by gene editing technologies and other pure parental cultivar line. And then we made a F1 hybrid and then evaluated the GABA accumulation in this F1 hybrid line. And as you can see here in this slide, this white bar show the control, and these two gray and the black bar show the F1 hybrid, which we used we used the gene editing line as a parental one of the parental line. And then as you can see here, we measure the GABA accumulation in the mature green stage and the breaker stage and late life stage where we are harvesting the tomato fruits. And then as you can see here, the tomato fruits at the late life stages show a higher GABA accumulation, which is 4 to 5 times higher GABA accumulation than the old control line, and which contain about 200 milligrams per 100 gram fresh weight of tomato fruits. This amount is enough to get the expecting function of this GABA. The 20 milligrams is enough to 10 milligram to 20 milligram is enough to get reduce the blood pressure. So that's why this amount mean that if we take maybe one cherry tomato fruits is enough to get the expected function, like lowering blood pressure and improving sleeps. Okay, so here up to here, this is a proof of concept experiment, and now we established the technology to improve GABA content in the tomato fruits, and then we start the actual production development of a commercial high GABA tomato and its commercialization. And for maybe everybody knows that the CRISPR-Cas9 is a patented technology with the patent is held by 3 different institutions. To make a commercial cultivar, we need to get the license from those patent holders. So I'm primarily belong to the university as a professor. So, but in Japan, the university cannot get a commercial license from the company. So so that's why I developed the University of Tsukuba UT spin-out venture company. The name is Solanaceous, which is 2018. And then we started the development of actual commercial cultivar, the collaboration with these companies. And here you can see some situation of patent for CRISPR-Cas9. The CRISPR-Cas9 is the patent is 3 patent is maybe responsible for the CRISPR-Cas9 technology, which we are going to use in the breeding. The basic patent 1 is MIT Broad Institute, and basic patent 2 is UC Berkeley, and also basic patent 3 is Vilnius University in Estonia. So the patent is a little bit complicated, but fortunately, in the application in the plants. These companies, US company, the DuPont, now is that the agriculture part is changed to the name is Corteva, and this Corteva accumulate these 3 patents. So then just we need to discuss with Corteva for the licensing. So then our company, Sonatic Seed, discussed with Corteva, and then succeeded in getting the patent from the Corteva for for use CRISPR-Cas9 technology for commercial cultivar development. And using, so that's why then we developed the commercial cultivar and using a CRISPR-Cas9 technology. And we started the commercialization on May 12 this year, actually, in the 2021 in Japan. And our tomato cultivar is, the name is Sicilian Rouge High GABA, which is the first gene-edited crop commercialized in Japan, and also which is the first gene-edited crop produced by commercialized all over the world, which is produced by CRISPR-Cas9 technologies. But anyway, so we started the commercialization in Japan. Okay, in Japan, I would like to somehow introduce the current situation of regulation in gene-edited crop in Japan. For the commercial use of gene-edited crop in Japan, we have to notify our products. Developer have to notify it to the 2 ministries, which is MAF, Ministry of Agriculture and Food and Fisheries, and Ministry of Health and Welfare. For according to the Japanese system, the developer have to do a pre-consultation with information about their products, and then they notify that they provided, they submitted those information to these 2 ministries. And for getting permission for cultivation, we have to provide the information to a MAF, and then also we have to get for food to use as a food, we have to submit it to a food safety information to a MHLW, and also for feed, we also have to provide the information to a MAFF. So during the pre-consultation, we have to create several amount of data which have to be submitted to those 2 ministries. Okay, I will show you some what kind of data we need to submit to notify the ministries. But before explain what kind of data we have to submit it to a government ministry, maybe I think that the audience today is not a specialist in gene editing or not. So here you can see, I'm briefly in this slide. I'm briefly introduced the category type of gene editing technologies. So far, I think the gene editing technology divided into 3 groups. One is SDN1 and SDN2 and SDN3. And some ministries say type 1, type 2, type 3. It's the same meaning. And in Japan, SDN1 just cutting the double-stranded DNA, and then the repairment is naturally repaired, and then that's resulting in the deletion and the insertion or base changes in the gene, and those changes become a mutation, which is similar to the natural mutation or artificial mutation induced by chemical mutagenesis or physical mutagenesis like gamma ray or a neutron ray, and so on. And so those crops produced by SDN1 is categorized as a non-GMO, and that means without a Cartagena protocol. And SDN3, which is a technology to introduce a new gene to this cutting site. So the produced mutants produced by this SDN3 is now categorized as a GM in Japan. And the SDN2 is a technology to introduce a small mutation by introducing the fragments prepared in the outside of the cell, and then put the small fragment to repairing process, and then that's resulting in the small replacement of gene. And these products produced by SDN2 is sometimes categorized as GMO and sometimes categorized as non-GMO. So it's a kind of a gray zone. Maybe this situation is same in other country already established regulation rules. But anyway, okay, so this is a category, and our products produced by SDN1 technology. So that's why our products going to be categorized in the non-GM. Okay, so here this slide shows that sometimes I use this slide to simply explain what is the difference of GM crop and the GE crop and the GMO. This is the number of the 34,000 is the number of the gene in tomato, and in the gene editing for engineering the target gene, we put the one genes, but in the final products we deleted this one gene, so the gene number is the same. But in a GMO, and if I introduce the insect resistance, then we put the one genes, and then the final products it contain one more genes, as you can see. And so these products categorized as the GMO. And okay, so this is a flow diagram of handling of food derived from gene editing technology in Japan, which is a flow by Ministry of Health for food safety assessment. And so developer need to prior consultation before the notification to the notification. Then a developer have to get a consultation with a ministry, and then during the prior consultation, then developer have to submit a several amount of data, which I show in the following slide. And then once this ministry decided to, which is not which is produced by SDN1, so that falls under the notification processes. And then developer formally submit the data and to notify their products to the Ministry of Health and Welfare. And then after this process, the developer can use the gene edited crop as a normal food. So this is a process of regulation of food safety in Japan so far. Okay, and these are points to be noted regarding the modification. This is MHLW. The for food derived from genome editing technology subjected to a notification, a developer set to notify the MHLW of the following information, and the developer have to provide several information: the name of item and the breed and the summary, and of the development of food, and the method of gene editing technologies used, and the detail of modification. And in our high GABA tomato, we use the CRISPR-Cas9 technologies, and then our target gene is a GAD3 genes. And the information on confirmation of that there are no remaining foreign genes or their parts. We need to prove that the foreign gene is not included in the products. So in our case, we prove that prove that this no remaining of foreign genes by genomic Sasan analysis and also a PCR, two methods. And the information on confirmation that confirmed a change in DNA do not cause the products of new allergen having an adverse effect on the human health or increase of non-toxic substances and contain. In this case, we use we did some bioinformatic analysis for allergen. It's like almost the same with a GMO, and also we measure the toxin content. As As you know, the tomato contain natural toxin, the name is tomacin, and then we measure the tomacin content and also related natural toxin in tomato. And for items which modified affecting the metabolic systems was performed in order to increase or decrease the specific components, informational changes in major components. We increase the GABA content, and the precursor of GABA is glutamic acid. So then we also measure the glutamic acid content in the products and provided the information. And also then we need to, if we have idea, then we need to report when we are going to start the commercialization. So these are information we have to provide it to the Ministry of Health and Welfare to get get permission to use our gene edited crops as a food. But this process is not so heavy like a GMO, and so maybe yeah, not so difficult. And also the we need to submit similar data to get the permission for cultivation to a MAF, Ministry of Agriculture and Fair. Then we have to submit these 8 items, almost the same items with Ministry of Health and Welfare. Okay, so and we provided this information to the Ministry of Agriculture and Food and Fishery, and then we got our products categorized as a non-ST produced by SDN1 and categorized as a non-GM crop, and then we can grow it in as a normal tomato plant anywhere in Japan. So, and we here you can see the notification with the certificate of. I'm sorry, these are Japanese, but this is a certificate for the completion of notification to the both ministry for environmental safety information to MAF and the food safety information to MHLW and the food safety information to MAF. So then after that we can first gene-edited tomato now we can use as a normal tomato. And for the labeling system in Japan, and okay, so I have a okay. So labeling system in Japan, it is not a mandate. We don't need to, developer don't need to make a labeling, but our Solanaceous seed company decided to give a labeling because of this is the first products in Japan and of GM gene edited crop. So that's why we decided to make a labeling as a first products. And then this company, Solanaceous seed, give this kind of a label, as you can see here, including the information of this tomato has been improved by gene editing technologies, and the notifications to MAF and MHW has been completed. Then we are going to provide this information to the user or consumers. Okay, so and then after this notification, this company, Solanaceous seed, decided to initially provide the free monitor seedling samples of Sicilian Rouge High GABA to the home gardeners, and which is November 11 last year. And after the announcement of these free monitor samples, we had a lot of requests, applications, more than we got 5,000 applications all over Japan. And that is amazing number than we expected. So because that means indicating that many Japanese consumers interested in these new products. And then we decided to start delivering the seedlings in this May 12. As you can see here, this is one packages for one applicant. We provided 4 seedlings and with some fertilizers, as you can see here, and then and some textbook how to grow these tomato seedlings. And then now many home gardeners, as you can see here, I'm also growing this tomato in my garden, and as you can see here, many I think almost 5,000 gardeners in Japan is now growing the gene edited tomato, as you can see here, and we could harvest start harvesting the fruits from the end of June, and we enjoy. Those gardeners now enjoy the tasting these gene edited crops. Okay. And finally, take-home messages. And okay, so here, I would like to summarize how we spent to develop a high GABA tomato during last year. Okay, so we started the basic research to find out the target gene of gene editing. We started in 2005, and then we almost spent 10 years to find out the target genes, which is effective target genes to increase the GABA accumulation in tomato. And then we started the application of gene editing technology in 2015 to increase the tomato because of, we funded a national project too. And then we completed the POC experiment research in 2017, and simultaneously we started the development of a commercial cultivars. And then we established a Sanatic seed in 2018 and 2019, because the Japanese government established the regulation rule in the 2019, and then we started consultation, prior consultation with the 2 ministries in 2019. And then we finished the consultation and completed the notification 2020, December 11. And then this year we started delivering seedlings to the home gardeners in Japan. And then now we are going to make some, in the next year, we are now trying to prepare some processed food using this high GABA tomato because of the request. We made some SNS group among the home gardener we provided the seedlings, and then we had a lot of requests to make some processed food using this gene-edited tomato. So then we are now developing a processed food using this tomato, and then going to commercialize in the coming year. Okay, so this is my final slide, and then I feel that now new era of rapid and precise breeding via gene editing. So gene editing allows us an order-made breeding for sustainable food production. Okay, so this is the last slide. Thank you very much for your attention. That's all my presentation is.
All right, thank you so much, Dr. Divya. Could you please come and moderate the Q&A session?
Yeah. Okay, so Q&A. Okay.
I think we are still waiting for Dr. Divya. It's okay. I can start.
Yeah, sure.
Okay, perfect. Perfect.
Yeah, sure. So I was having some difficulty in looking at the Q&A. So
No
thank you so much, Dr. Hiroshi, for the wonderful presentation. So you have covered so well, the basic, the intent of genome editing for the health promotion is something that rings very well with all humankind. So it was great to understand the type of genome editing that is rendered non-GMO or GMO by Japanese government. And all the workflows you have covered so well in detail, from the pre-approvals up to the commercialization, all the workflows that needs to be done in this regard. It was a great understanding for us who have not yet got any exposure into such events. So from here, I will move on to the Q&A session. The first question is, what kind of regulatory mechanism does apply? What kind of regulatory mechanism does apply on commercialization of CRISPR-derived products, especially GABA-rich tomatoes?
I think that I already
covered. Yes. The second one is, did you conduct analysis to understand if the composition of GABA tomato has changed compared to its conventional counterpart? And is this required to achieve regulation status in Japan?
Oh, okay. So yeah, so I measure the GABA content in the yeah. So you conventional cultivars. Yes, that's it. I think it is required to the by the government because we measure the original GABA content of original cultivar, and then we provided those information to the GABA to the ministries.
Okay, thank you, Doctor. The next question is from Dr. Shidi. In terms of off-target effects often observed from CRISPR-Cas9 mediated gene editing, did you observe changes in other metabolites in the high GABA tomato? Are there any special considerations with regards to off-target effects for approval for food production in Japan?
Okay, so as I explained in the detailed slide in the by the Ministry of Health and Welfare, they requested us to check the presence of absence of off-target mutation in our products, and then we did some informatic analysis and also some sequence analysis for the potential off-target sequence. But and then we provided those information, and fortunately, our products we could not detect any off-target potential off-target mutation. So we provided those information to the ministry of government, and also the okay as also as I mentioned that we also provided information of the other metabolites in our tomato, like glutamic acid is a precursor of this GABA, so that's why we evaluated the amount of glutamic acid in the original line and our high GABA line.
Okay.
But the content was not significantly different between both lines.
Okay, thank you. Is all the information about gene-edited plants released into public? Do we release all the information to the, about the genome-edited plants into public?
I can't catch what's the meaning of this question clearly. Can you explain more detail about the
yeah, so what I understand is like the participant is asking, just like Dr. Shidi mentioned, that if there are any other changes associated with, editing the gene for a particular trait, if there are some residual effects or some other effects, are all those details disclosed to the public?
Okay, so we combined both traditional breeding techniques and also gene editing techniques to making these products. So during the evaluation, we compare the growth of the our gene edited line and with the original line. And fortunately, we could not detect any differences in the development. And also, we detected, we checked the stability, genetic stability of mutation introduced by gene editing technology for 3 generations, which is a similar technique with the conventional mutation breeding techniques.
Okay. Yeah. Thank you, Doctor. And the next question is there a quantification mechanism for how much of tomatoes to be consumed to reach optimum scale or range of blood pressure? Okay, so and for the GABA, there is a many experimental data for the effect on the human, and according to those information, the 10 milligram to 20 milligrams of GABA is enough to expecting the lowering the blood pressure. And so that's our tomato contain 1 milligram to 2 milligram of GABA per gram flesh weight. So that means that 10 grams to 20 grams of fruits, fresh fruits,
is enough to get the expected function. And also, there is a many tests for excess use of GABA. Now, if we can take 100 to 1,000 times of GABA, there is no effect on our human health. So GABA is a very well-studied functional material so far. And we, based on those information, our government decided it is not any harmful effect on or as a food.
Okay.
Yeah.
The next question is, what SDN2 product qualifies as GM and as non-GMO?
Okay, but okay, so actually the Ministry of Agriculture and the Fishery in Japan is there using a Cartagena protocol. That means that, as you know, that the Cartagena protocol is a process-based method, and then in the SDN2 we input a small DNA fragment for the repairment of the braid, and so that's so that means that the small DNA fragment, which is processed out externally processed in the cell, and then so that's why in the that is considered to be a GM. But in the Ministry of Health and Welfare, they are decided as a product base. They are not carrying the Cartagena protocol. So they are decided the product basis, and then so the mutation can be potentially found in the natural mutation, even which is produced by SDN2, that is categorized as a non-GM.
Okay.
But so this is a matter of still matter of discussion. So it's a kind of a gray zone.
Okay. Thank you. Next question is Senatec planning to market GABA tomato seeds in other countries?
Okay. Okay. Thank you very much for that comment. And of course, but initially we wanted to completely get acceptance by the Japanese consumers, and then after that, I know that the different countries now are trying to establishing regulation rules for gene-edited crop, and if that some country is completed the development of technology, the regulation rule, and then so we of course we wanted to export the GABA tomato or market in those countries.
Okay. The next question is Senatec paying any royalties for the CRISPR tool license?
How do you mean that? You mean that okay? As I explained that this CRISPR-Cas9 techniques basic patent is held by the Corteva in the US, so that's why the we have to pay some royalty to the CRISPR-Cas9. Yeah, we cannot freely use it.
Okay.
But this situation is a same situation in some other industries. Like a motor company using a lot of external license to develop their cars. Not so right. special issues, I think.
Right. So the next question is, can CRISPR or gene editing be applied to get out of tight linkage drags?
Mm-hmm.
So if a trait of interest, has some linkage drags, maybe supposing, let's say, if GABA tomato, regulating gene is closely linked with yield or the fruit size, which is not suitable for the market, can CRISPR, a gene editing tool, be applied to, get out of that tight linkage drag?
Okay, that's a good idea. Of course, that if we know that the two important gene is linked together, and then if we wanted to change the one genes, so then CRISPR-Cas9 technique is a very, very powerful tool. And
Okay.
actually, we are trying to cut the subtle linkage drugs in some traits.
Okay, that's good to know. That's really very, very encouraging, right? So the highest GABA accumulation was found in green maturity stage and gradually decreased in red ripened stage. What could be the reason for this decrease?
Okay, so during the ripening stages, the GABA T enzyme activity is increased. GABA T enzyme is a enzyme which can degrade GABA. But so that means that if we knock out GABA T activity during the ripening, we can keep the high GABA content. But unfortunately, the GABA T expressed in the all part of the plants. Then if we knock out the GABA T, the GABA increased in different part of the organs like the stem and the leaf and so on. That makes those organs become dwarf or small, and then it has a significant effect. But the reason why we choose a GAB3 gene as a target, because which is predominantly or only expressed in the fruits. So that's why we targeted GAB3.
Okay. The next question is, how many off-type mutations have you found during performing the CRISPR-Cas9 mutation experiment?
You mean that off-type mutation? What do you mean by off-type mutation?
Yeah, so even I, maybe the person who has asked the question can, type it with more detail. By the time I will move on to the next question. All right. So the next question is, could the speaker share advantage and disadvantage of GABA gene? If I yeah, so
maybe these are different questions. I think.
Yeah, it is a different question. So by the time I process the off-type, what is meant by the off-type mutations in the previous question, I just thought like I will just move on to the second question, and meanwhile you answer that, we will go back to this off-type mutation question. So the question is, what are the advantages and disadvantages of GABA gene? So we know the advantages for sure. Are there any disadvantages?
What is the disadvantages of GABA genes?
Yeah,
what
so
do you mean by
yeah, so
of GABA?
maybe one thing can be as other people who have asked question can be like, if we consume about some quantity, minimum quantity, maybe the blood pressure can go down, which can be detrimental, detrimental to a person with low BP. So one is that, are there any other such disadvantages associated with this particular enhancement of this of GABA in the tomatoes?
I think that maybe we have no effect on disadvantage of GABA, because we targeted specific genes which can regulate the GABA accumulation in tomato fruits, and so that's why there is any other disadvantage of GABA gene regulation in the tomato plants development.
Okay. So I think like the question about off-type mutation might be like, when this attempt was happening, when we were attempting this gene editing, did any other event happen which are of interest in tomato?
Okay, so when I described, explained in the part of potential of target mutation, and we check the potential of target mutation using a different kind of bioinformatic tools, And then we found some potential target of this CRISPR-Cas9 target DNA, and then we made a sequencing of those potential genes, but we could not find any mutation in such a potential genes. So that's and also we checked the absence of the unexpected effect on the plant development by during the progressing the generation, and then so we could not find any effect on the gene editing in other developmental processes.
Okay. So thank you.
Dr.
Yeah,
Divya?
yeah, it's time. Yeah, this is what I was saying. So thank you so much, Dr. Hiroshi, for being patient, answering all the questions. There are many more questions in the Q&A box, which will be answered personally. You can go back and check it. And from here, we move on to our next presentation. Once again, thank you so much, Dr. Hiroshi, for all the insights that you have given through the webinar today. Over to you, Shrishel.
Thank you, Dr. Hiroshi. It was a very excellent presentation, and for people who are into the plant breeding and also other domains in agriculture and also other biology discipline, this is one of the fascinating science, and therefore many people were keen on understanding the technical part of the gene editing. And thanks for the nice presentation. And when we know that the gene editing has a lot of advantages, and also it has found the commercial utilization in part of Asia, but still there are many countries and governments struggling with a regulatory status of gene edited crops. And there are many questions being asked, like, are they regulated under GMOs? Should they be regulated on their own? How to develop winning R&D and commercialize strategy for gene edited crops? And a lot of questions on the regulatory part, I think, and yeah, as soon as we talk about gene editing on the science front, and there will be a lot of questions on the regulatory part, and most of the questions are they do not have answer. And I think with that, I would like to introduce to our next speaker. Our next speaker is Mr. Fabrice. Is a patent attorney and a partner with the IP law firm Rouse, where he co-manages the global patent group. He has over 2 decades of experience in patent protection and litigation in Southeast Asia. He is a member of Standing Advisory Committee before the European Patent Office and a consultant to the World Intellectual Property Organization on biodiversity. Fabrice is a researcher on climate science at the University of Edinburgh Carbon Innovation Center, and he is the authority when it comes to the patenting in a biological discipline. And I would like to request Fabrice to take us through the presentation. Floor is yours, Mr. Fabrice.
Thank you very much, Shrishel. I really appreciate. I'm trying to turn on my video, but for some reasons, is it that I can't for the time being? So maybe that will start my video. That should start. Okay, wonderful. Yes. So hello, everybody. It's a great pleasure and honor to participate to this webinar. Thank you very much to APSA for their invitation. And the topic, matched so well with what Dr. Ezura-san just, covered a few minutes ago. It's a great transition, and it shows that science and law or regulations, can be combined and even explained by non-lawyers. Dr. Ezura-san just perfectly achieved. So my talk, and I will share my screen with you right now, but my talk will cover basically, I hope you can see my screen right now. Can you do that?
Yeah, please go ahead.
Lovely. Yeah, my talk will cover basically the legal environment in relation to the GMO versus the gene-edited organism. And I will also share some of my experience as a patent attorney in relation to the patentability of gene-edited process or product. Dr. Ezura-san has explained much earlier that there are many patents and it can be quite complicated due to the licensing structure to acquire or secure rights to exploit the CRISPR technology. So let me start with basically quickly the agenda and the summary of our presentation. First, we will briefly go through the introduction on gene editing versus gene modification, and I don't pretend to give extra information that's the one Dr. Ezura-san just gave, but from a legal perspective, I will maybe give you some perspective. It's also very important to contextualize gene editing in the world we are evolving at the moment, at the policy level especially.
Excuse me, Mr. Fabrice, sorry to interrupt. Could you please enter the slideshow mode so that it's easier to view?
Is it better?
Still waiting? Yeah, perfect. Thank you so much.
Thank you very much. Thank you very much. Then there will be an executive summary, in order to just, put in brief words what my presentation will cover, so that can be shared with your colleagues or friends easily. And then I will cover the IP protection strategy, as I mentioned, and also the regulatory challenges, whether gene edited is regulated as a genetically modified organism, and some countries have taken this view, for instance, the European Union very clearly, or New Zealand, or is it regulated as gene edited as such? For instance, US has taken that perspective, and in the middle you have countries like Japan that apply a case-by-case basis, and there will be takeaways. I'm aware that probably most of you are not lawyers, so I will try to use words that are not too legally, and definitely if you have any concern questions, I understand there will be Q&A at the end and we can cover anything. So again, introduction to gene editing, CRISPR versus genetically modified organism. So as it was explained earlier, gene editing is different from gene modification because there is no introduction of a foreign gene. It's just a matter of modification, enhancement in order to achieve a particular application. It is said, and this is a bit a concern to us as a lawyer, that gene editing essentially allows to edit gene in the same way that happens in nature. And to me, that's a matter of concern, because when you talk about patent protection, for instance, we know that patents cannot protect something that exists in nature, like a plant, like an essentially biological process. So when we start saying that gene editing is the same as what happened in nature, we run a risk that eventually the patent office, when we apply for protection of a gene edited, they say to us, well, you can protect it because this is the same as what happened in nature. So maybe that's the added part to what Dr. Ezura-san just mentioned. So the context, what is the context of gene editing? Probably a couple of years ago, if I had to make this presentation, I would have had two main contexts, the IP protection, because this is relevant to companies when they do research in gene editing. They hope to be able to harvest and capture some IP protection through patent, trade secrets, and so on. And regulatory market access, you want to be able to commercialize eventually your product or do further research and development. But recently, climate change has reached level of awareness that never happened before, and for good reasons. And it's an opportunity. Why is an opportunity? Because several reports from the United Nations, in particular the latest one released a couple of days ago, the what we call the sixth assessment of the Inter-Panel Government on Climate Change. This report mentioned that in order to adapt to climate change, and this is the adaptation strategies, right? In order to adapt to climate change, there are various solutions, and one of them is gene editing. Basically make, for instance, rice more resilient to flooding or extreme temperature and so on. So we now have actually a support from the United Nations official body saying that unless we develop gene editing, it will be complicated and challenging to address climate change in terms of food supply. And why is relevant? Because some of you may be attending this corner from the policy level. When we lobby changes of laws, when we lobby government to amend their regulation from saying, oh, gene edited must be regulated as a genetically modified organism, for instance, and we want gene edited to be regulated separately or differently from gene modified organization, we need solid arguments. And with this climate change dimension, we are now able to say, well, the world needs gene edited crops. And there is time pressure. So we can't wait 5 years, 7 years for the approval, as it happens sometimes with gene modified organism, or sometimes gene modified organism are simply not allowed to be commercialized. So I see it as an opportunity, and I wanted to raise it in terms of positive context. This is an interesting study made in Vietnam, where the authors interviewed local farmers producing rice, and they asked them what would be the best manner to adapt to climate change. So you see different options, you know, soil conservation, maybe because the soil is saturated by CO2 and so on. But they have, you know, definitely the option to modify or enhance, I guess, the crop varieties. And it is one of the elements that was identified by the local community. And we are talking here about local farmer. It's not, you know, the large scale corporation or big, companies. We are talking about, you know, small size, you know, 5 to 20 people working in a farm. So that's very, very revealing of the needs for gene editing at the local farmers level as well. And for that reason, if you look at the growth projection, gene editing is picking up. You can see a breeding, of course, you know, remain fairly stable. Genetic modified organism had their glory years when it was where it was possible to commercialize. And gene editing is now, you know, definitely picking up. And I think the climate change dimension, the increase of population play a key part. So what are the issue, the main issue here? Well, the main issue are government need to have an appropriate regulatory system and guidelines to address two main issues. First, of course, the need that I explained earlier in terms of food supply, in terms of ensuring regular and stable yields and production, but at the same time, the safety nets. And then we have this debate that was already introduced by Dr. Ezura-san a few minutes ago, debate whether CRISPR or gene editing is regulated as a GMO or not. Should we apply GMO label on gene-edited products, right? Definitely, you won't be surprised, the seed industry really want to see gene-edited product excluded from the GMO legislation, right? So basically to be treated the same way as conventional breeding. And from the patent side, well, from talking with clients in this industry, they also want to be able to patent gene-edited, either the product or the process of making a particular gene-edited fruit or, tomato, for instance. But as I mentioned earlier, it seems we are going to two opposite direction, and to policymaker, it's important to raise the following risk. When we say that gene-edited crops are not gene modification, our main argument is to put forward that gene editing essentially allows to edit gene in a way that happens in nature. That's what I mentioned earlier. But when we say that gene-edited crop should be patentable, as the industry push for that, we raise here a risk that the patent office will tell us, well, you just mentioned that gene editing is the same as essentially biological process, and we are sorry to say that essentially biological process are not patentable. So we have to be very, very careful when we promote the idea of standalone gene editing legislation for approval and commercialization. And when we say that gene edited need to be patented for the reason that I mentioned to you, since there is a risk that the patent office will investigate and conclude that this is an essentially biological process. So what is the executive summary of my talk? There's many points, but I can summarize it. Basically, there is lack of consensus between countries in terms of how genetic edited crops and so on are regulated. Some countries believe that it should be regulated as GMO. Some countries believe it should not be regulated as GMO. So this is a lack of consensus, and it is a problem for companies because you need to navigate through many different systems. Interestingly, even though you have a legislation in place, for instance, that was the case in New Zealand, the courts may amend or force the government to apply a different procedure. And for instance, New Zealand for many years say, okay, genetically edited crops don't need to be regulated under GMO. It's not a requirement, but the courts overrule, they change basically the approach of the government, and now gene-edited crops need to be regulated under GMO. Some countries, some regions, Africa, Southeast Asia, don't really have a comprehensive system, and that's difficult. It creates a lot of legal uncertainty. Interestingly, China, that has a lot of patents in relation to gene editing technology, well, don't really have a regulatory framework to approve, commercialize gene-edited plant. There is also in Northern Europe a very strong sense to deregulate gene editing. In other words, some countries, Norway, Switzerland, nowadays the UK after the Brexit, really want to remove gene edited from the GMO legislation. And finally, we can see, as always, when dealing with legislation, a big gap between the regulatory and the science. Basically, things are moving very fast on the science side, but on the legal side, it takes time. So we are always behind. Now, this is a summary of the basically various legislations. So you have countries or region that we cover briefly today. Most I mean, you know, with very few exceptions, but most countries in the world, we protect gene editing through either patent or trade secrets or plant variety. So it's not a big concern to you as a researcher or owner of this technology to protect it. Where the differences are is, as I mentioned earlier, how this technology, how the product or process are treated when it comes to research or commercialization or labeling. Is it GMO or is it gene edited? And there are, as you can see, a lot of ongoing policy development towards deregulation. In other words, there is a positive trend trying to motivate and encourage government to remove gene edited from the scope of the umbrella of gene modified organism legislation, which is very encouraging. Now, quickly on IP protection strategies. Well, basically, there is a specificity in patent protection or IP protection for plants. It's very different from mobile phone technology, for instance. If you look at what Nokia invented 20 years ago, I bet you that LG, Samsung, Apple do not rely on Nokia past technology to do mobile phone. There is no continuity because it's a very, I would say, you know, highly innovative industry, which come with new application, new products. Therefore, you don't really rely on old technologies. Plant is slightly different. There is actually a level of continuity in terms of innovation. And if you look at you know, this graphic here, we are comparing different innovation cycle. We are with plant and gene editing in the bottom one, the high-paced innovation. So basically, innovation is fast, but it relies a lot on existing innovation. And that's what Dr. Ezura-san mentioned earlier when he said, well, we need to go back to those 3 key patent owners in order to be able to commercialize our invention. Even though we created something new in Japan, we still need to embrace other people's technology to commercialize it. So this is what we call the cheating effect of the legal uncertainty, because you need basically to assess who owns what in order to research or commercialize your potential gene-edited crops. And that's the task usually of the R&D team together with patent attorneys. There is also another issue when we look at the patent or IP protection, is that there is no real harmonization. Different legislation, different ways, different exemptions, and I will show you some examples here. Even though the majority of the World Trade Organization, so 165 countries in the world, agree that we cannot obtain patents for a plant or essentially biological process, in other words, something which happened in nature, we should be able to obtain a patent for modified organism, including gene edited, because there is a human intervention. What Dr. Ezura-san did in his lab with his team members, I'm sure, you know, required a lot of research, a lot of discussion, and then human intervention in order to edit the particular gene, enhance characteristics, and that's usually patentable because you can differentiate from what happened in nature, even though the final product is the same, but the process has involved a human intervention. Now, what is very interesting, and that's very recent, the European Patent Office, the only office in the world, has made an interesting disclaimer request. Basically, they say the following: if the gene editing results in exactly the same technical features as the one in the natural product, then you need to disclaim your patent. So when you file a patent, you have what we call claims. So independent claims, you know, normally very broad, and you will say, okay, you know, this is the scope of your invention. You describe your invention and you tell the public or the world what you want to own based on your research and so on. Now, if that particular patent, the features can be achieved through natural plant, naturally grown plant, or essentially biological process, you will have to issue a disclaimer. And that's very, we say, impacting your client's rights, because once you issue a disclaimer, you no longer have exclusive rights in this particular feature. And I wanted to highlight that. Maybe there will be further questions during the webinar, but that's an important development. So basically, to reassure everybody, you know, this technology, the gene editing technology itself, is patentable, and we have seen many patents in the world already, and also the product achieved from the technology is also patentable. But patent is not the only protection. You may consider plant variety protection. That may be possible in some countries where new variety, so basically you may claim that a gene edited plant is a new variety and then qualify for protection. Some of our clients basically don't want to apply for patent or plant variety because they want to keep it secret. They still want to license and commercialize, but they want to keep secret the technology behind the editing, so no one will know how they did it. So here you can see comparison. I won't go into details, but they definitely not the same. And I won't say that patent is necessarily better. It just depends what you want. But patent is normally more expensive than plant variety protection, and definitely than trade secrets, because trade secret, you don't need to register it. The filing strategy, well, you know, you need to identify the countries where you want to protect your gene edited crops. You need also to consider the type of IP rights. As I mentioned, you want to go for patent, you want to go for trade secrets, you want to go internationally, so covering a lot of country or just your country. There are different elements to consider. So let's look at what is currently protected. There are two interesting studies because they don't find the same numbers, but we basically agree with the second study because we conduct ours. So basically one study say there are about 7,500 patents covering CRISPR around the world. That's quite a lot. And another study say, well, it's about 2,200 international applications, and we found almost the same data. So I will tend to rely more on the second, you know, data that I'm sharing with you. This is more or less the key owners. You can see, you know, the number of patents they own. You can see that there's been a real peak in terms of publication of patents in years between 2010 to 2016. That's very much where CRISPR was highly being developed. And
Mr. Fabrice, sorry for the interruption. We have 4 minutes to the presentation.
Okay.
It's an interesting subject. Please, could you?
Sure. I will then move to the second part quickly. This one. Yes. Okay. So basically, I will cover now the second part of my presentation, which is whether or not the genetically modified organism will be regulated as GMO. So whether the gene edited will be regulated as GMO or as gene edited as such. So very quickly, Australia, they decided that any gene edited crops will be regulated as gene edited. So GMO does not apply. Japan, as Dr. Ezura-san has explained, basically they apply different scenarios, SDN1, SDN2, SDN3. And if you fall under SDN1, you're not required to pass through the GMO legislation. USA has made very clear statement that they protect and cover gene edited as such and not GMO. Latin America, very similar to the US, so pro gene edited. Israel as well, and that's an important country when it comes to plant research. UK is leading to that direction as well. They really much, after the Brexit, want to take a different position from the EU, so they are pro gene edited standalone legislation. Canada is a bit unique in the world. You know, they have a system whereby they is very much product based, so it's a case by case, but they give some criteria. You know, if the new plant variety has traits that have 25 to 30% higher than the commercial variety, then it will be considered as GMO and then be under the GMO law. India is definitely moving to a similar system as Japan, but
Mr.
this is still
Fabrice,
under discussion.
sorry
Yeah.
for interruption. We don't see much questions. I think maybe, you know, you can take 10 more minutes. Please go ahead.
Okay, thank you. And then on China, as I mentioned earlier, has many patents covering CRISPR, but as of now, their legislation does not really exclude gene edited from the gene modified organism. So therefore, it is still unfortunately covered by a GMO. EU has made very strong decision back in 2018 that gene edited are covered under GMO. And that's very much seen as an obstacle for the industry in Europe. And they've been lobbying activities, as you can see, before the EU Commission in order to amend the legislation and allow gene edited to be separated from gene modified organism. New Zealand, well, it's a bit a surprise here because the country has changed its legislation from gene edited supportive to nowadays gene edited as subject to gene modified legislation. Korea is still work in progress. They haven't, to my knowledge, there's not been any firm decision to regulate gene edited separately from gene modified organism. And the ASEAN, well, the ASEAN has, we say some countries, Philippines, have tried to very much push for a more ambitious legislation to cover gene edited as such. But the rest of the ASEAN countries are still very unclear as to what should be the individual countries' position. On a regional basis, yes, there is a plan for action 2018-2025 that, you know, cover GMO, but also, as mentioned, gene edited. But at the moment, there is no clear direction as to whether gene edited should not be regulated under GMO. This is for information. You have details on the Philippines and the ASEAN countries specific legislation. So my last slide, well, I can say that there will be, in my view, further countries supporting the indigenous legislation of gene edited crops, similarly to what US or Australia have done, and I think that's a positive. There is also an increased consumer acceptance. I think as long as labels are clear, I think we always talk about social acceptance of, you know, genetically modified organism, but we should remember that for many years, you know, legislation were very poor on labeling, and I think as long as labeling is clear and transparent, social acceptance is always better. The risk, if there is no harmonization between those countries in terms of gene edited labeling and approval system, is that you may have trade issues. So some gene edited products, crops, and so on, or food cannot be exported to countries, which will create a barrier. And I think that's an issue that should be taken at a regional level. And finally, intellectual property, as I mentioned, this gene edited technology is protected through patents, and we can expect a large increase of patents in coming years. It is estimated by 2040 that there would be an increase of 80% before the European Patent Office of gene-edited patents for particular crops. So that's the end of my presentation. I'm sorry I had to rush towards the end, but very happy to take on questions.
Yeah, thank you, Mr. Fabrice, for taking us through one of the very interesting topic that I think most of the breeders and also people who are involved in the biological science would like to know and also understand the progress in this area. As you have mentioned, I think there is no doubt that this is one of the fast-paced moving innovation among all the disciplines. And definitely, I think, the global regulatory need to come up with a homogeneous regulation on this part. And, we can see a ray of hope from Japan, one of the Asian countries.
Yeah.
We can see the commercial success of the GABA tomatoes. And definitely, I think, many countries will look up and also follow the country. And also good news is that I think few countries, few of the European countries are, coming up with the disclaimer. And that's a good, thing for the gene-edited crops. And major part of the Europe is putting this as a GMO. That is one of the, the really, one of the concern for people who are working on this one. And with this, I would like to open the forum for Q&A, as it is, one of the gray area. We have a few questions, and I will start with the question from Jiofeng. And the question is, based on your experience and understanding, do you have any opinion as to whether Thailand will or will not regulate gene-edited similarity or different to GMO? This is the question.
Thank you very much. That is a question that resonates very well in the context of what we have seen, for instance, in the Philippines, as also an agricultural country, of course. The Philippines has taken a quite, progressive view in terms of gene editing by, using words in their regulation that seems to indicate that they differentiate the two when it comes to approval procedure, even though, the term or the word could have been selected better, but, it's an important step. I would say that Thailand, as an agricultural country, has quite, I would say, progressive when you see what hemp and cannabis legislation is, achieving at the moment. I think there are hope for the country making that difference, and especially in terms of potential for exports as well. I think, domestic consumption is one thing, but export is another thing. And as I mentioned, with climate change element, we may need this sort of liberalization of the legislation. So yes, I would say there is potential.
Okay, thank you. I have one question around this. As you have mentioned, there's no homogenized policy, when it comes to the gene-edited crops. And this is being discussed in the United Nations as one of the strategy to come up, overcome the climate change, right? And are there any efforts going at a UN level to bring all countries together to come up with a common policy?
Yes, absolutely. I think first of all, the policy makers at the UN at the moment are very much driven by adaptation strategy rather than mitigation. And in terms of adaptation, in the field of agriculture, you don't have too many options. And I think that is why gene editing is at the moment capturing so much attention and appetite from countries to just address an urgent issue. So they no longer have choices, I would say. So yes, there is a strong consensus. Yes.
Okay, that's a good sign. We have other question from Korea Seed and Variety Services. Is it necessary to check the safety of the product where the GE tools are used in developing the crops?
Yes, I would say that even in countries where the gene edited are regulated separately from gene modified organism, there is actually a safety assessment that is much less burdensome as for genetically modified organism, but they are not free from, of course, checks and investigation. I think it's quite similar to what Dr. Ezura-san just mentioned earlier for Japan. It's not because they are SDN1 that they were removed from any safety test.
Okay, thank you. And audience, please feel free to type down your questions. We have 10 more minutes for taking a few more questions. Okay. And if there are no questions, Mozam, shall we move to the next part of the session?
Yes, Mr. I think there is one more question. Yeah.
Okay, good. We have a question on India has banned GM food crops, unfortunately. Consuming GM cotton byproducts as seed oil and cattle feed indirectly. Control over market is difficult. Okay. India has GM food crops, unfortunately. The question is about banning the GM as a food crops in India. Therefore, I think the question is, it's very difficult to control once the event becomes public. This is a question. Basically, it's a comment.
Yeah, it's a comment. Yes.
Yeah. And if there are no questions, I would like to thank all the participants for attending this session, and also thanks to our honored speakers, Dr. Hiroshi, who has taken us through the technical part of the gene editing, where GABA tomato has been a commercial success in Japan, and that's one of the success story. And also he has covered a lot of technical things about types of genome editing and what are the categories. The type 1 comes under non-GMO, type 2 comes under GMO and non-GMO based on the event. SDN3 is a GMO, and also he has taken us through the legal approval process in Japan, and it was a very nice presentation. And thank you, Dr. Hiroshi. And coming to the second presentation, Mr. Fabrice, it's one of the complicated area where the gray area, where a lot of discussion and debate is happening. And thanks for taking us through the legal status in the different countries, and also thanks for taking us providing an executive summary happening in the different countries. And also I would like to take this opportunity to thank all the participants for their patient hearing, and also keeping the Q&A session very active. Also thanks to APSA for keeping the web series going on even during the pandemic situation. Thank you one and all. And with this, I would like to request Mozam to take us through the webinar series 6 and also its update. Over to you, Mozam.
Yeah. Thank you so much, Mr. Sushail and Dr. Divya, for moderating the session today, and also to both of our speakers. It was really informative presentations from both speakers and from the audience as well for participating in the Q&A. So I'll just take 5 minutes to update for the next webinars, and also for some of the information that we should share with our attendees. Yeah, so as you can see on the screen, so webinar 6 for the ASCRT series is scheduled to be on October 27, Wednesday, and more details will be shared with you very soon via email. So the speakers and also the theme of the webinar will be shared very soon. So please stay tuned for that. After that, we see a few comments in the chat about the presentations. Yeah, sorry, this one, yeah. So all the attendees of today's webinar will be automatically registered for the next webinar, as it happened for the previous one. So all of you who attended today are automatically registered, and you will receive your specific username and password before the next event. So you do not need to register again, but yes, if you have any of your colleagues who are new and who would want to register for the next webinar, you can do it via our website, and we'll be sharing those details with you very soon. Next is the post-webinar survey questionnaire. So this is very important for us, and also I believe that it's important for the attendees as well to submit their responses. So after this webinar, you will receive an email from the Secretariat. It will take 3 to 5 minutes for submitting that questionnaire. So all those who submit the response to the questionnaire will be given an e-certificate of attendance. This questionnaire is really helpful for us to improve our webinars and also to arrange next webinar series. So please make sure to respond to the questionnaire. Next is the new initiative from EPSA, which is the training courses for our members. So all the attendees from webinar 1 to 4 who have attended our webinars before already received an email from the Secretariat requesting to respond to this questionnaire. So this is basically a new initiative where we are collaborating with different academies and institutions to prepare custom courses for our members. So these courses will bring up-to-date information for the employees of our member companies. So make sure to respond to this one as well, and we'll update you once we receive the responses from most of the members. And those who attended today's webinar for the first time will also receive this questionnaire. So this is also requested to please respond to this questionnaire. Next, we received some comments about the presentations today. So both presentations and the recording of today's webinar will be shared with you by tomorrow. So you can access that. And recently we received some comments about the previous presentations, recordings or Q&A. So I'll just be taking you through how you can attend the previous EPSA events. So if you attended the event, you would require your login ID, which is specific. And if you have forgotten your login ID or password, you can contact Mr. Bobby from EPSA Secretariat. His email is given on the screen. And if you did not attend a specific event, but you would want to have access to the recording or presentation, you may contact to the company representative and you can log in through that. So the procedure is simple. You go to EPSA website and you click on Events tab. Once your Events tab, you click on Past Events. On the past events, you click on the event that you are interested in. In this case, it's showing on the ASCRT4 recording and the details. So once you click on this one, it will ask you to put the username and password. So you can put your username and password if you attended, or you can put the company representative's username and password. So once you put that, you will be signed in and you can access to the presentations, recording, and the Q&A files. So with that, I would like to thank everyone for attending today and making your time. Thanks to R&D Advisory Group for making sure that this series continue and bringing the interesting topics. Thanks to both speakers for their time today and the Secretariat as well. So I wish you all have a good day and we will see you in the next webinar.